Hello everyone, welcome back to my channel. Today I am going to tell you some interesting facts about why healthy young children usually don't get severe COVID-19 disease. Throughout the pandemic, there have been proportionally fewer recorded cases and deaths from COVID-19 among children, adolescents, and young adults, although their tendency to experience fewer and milder symptoms may also have resulted in fewer tests being done. Children experience lower infection rates, the symptoms are typically milder, and they also appear to transmit the virus less and are not primary drivers of community transmission. Children have trained immunity, which involves epigenetic reprogramming of innate immune cells, including NK cells, with their rapid ability to kill virus-infected cells by perforin and enzyme degranulation. After exposure to certain stimuli, including infections, these trained cells react faster and more strongly to subsequent pathogen challenges, providing enhanced protection. A study published in JAMA found that 98% of babies born to mothers who received an mRNA vaccine during pregnancy had detectable levels of antibodies 2 months after birth, and 57% of babies had detectable antibody levels 6 months after birth. Another report published in MMWR found that babies born to mothers who received 2 doses of mRNA vaccine during pregnancy were 61% less likely to be hospitalized with COVID 19 in the first 6 months of life. Cytokine storm is an excessive immune response to the infection that is associated with widespread tissue damage and high cytokine production. Cytokines are vital to the pathophysiology of COVID-19. Uncontrolled generation of pro-inflammatory cytokines contributes to acute respiratory distress syndrome that results in elevated plasma levels of interleukin 2, 6, 7, beta tumor necrosis factor, and interferon. Children tend to have reduced inflammatory responses to SARS-CoV-2 infection. Children infected with SARS-CoV-2 often have co-infections with other viruses, including commonly circulating HCOVs. These viruses could interfere with the replication of the SARS-CoV-2. Frequent recurrent viral infections could also induce an enhanced state of activation of the innate immune system, including epigenetic changes in the trained immunity, making it easier to clear SARS-CoV-2. Children might be protected against SARS-CoV-2 as a result of pre-existing cross-reactive antibodies from more frequent and recent HCoV infections. Preliminary data show that while antibodies to HCoVs cross-react with the spike protein of SARS-CoV-2 and SARS-CoV, these antibodies are rarely neutralizing, as they do not bind to SARS-CoV-2 receptor binding domain. The same is true for cross-reactive T cells to SARS-CoV-2, which are also present in high numbers in the elderly and might be detrimental. Another potential explanation for the less severe manifestations of COVID-19 in children are the differences in their microbiota. The microbiota plays an important role in the regulation of immunity, inflammation, and mucosal homeostasis, as well as in the defense against pathogens. Children are more heavily colonized with viruses and bacteria than adults, especially in the nasopharynx, where microbial interactions and competition might limit the growth of SARS-CoV-2. Melatonin has anti-inflammatory and antioxidative properties through several different mechanisms. Melatonin protects against ARDS and hemorrhagic shock during viral infections, and it also inhibits calmodulin, which increases ACE2 expression and retention on the cell surface. Melatonin secretion is negatively correlated with age, with particularly high levels in infants and healthy young children, which could contribute to the mildest symptoms in this age group. Many live vaccines have off-target immunomodulatory effects beyond protection against their target disease. BCG vaccine influences the innate and T-cell immunity by epigenetic reprogramming of immune cells and by altering cytokine responses. As children have generally received BCG and other live vaccines more recently than adults, it has been postulated that this contributes to age-related difference in COVID-19 severity. Viral load influences the severity of COVID-19, suggesting that lower intensity of viral exposure might be another factor leading to less severe disease. As children are usually infected by an adult, they are infected by a second or third generation SARS-CoV-2. For SARS-CoV and MERS-CoV, subsequent generations of virus were reported with reduced pathogenicity compared with the first-generation virus. Children have a lower prevalence of the comorbidities that have been associated with severe COVID-19 in adults, such as obesity, diabetes, hypertension, and chronic kidney, lung, and heart disease. However, interestingly, even children with serious medical conditions who are on immunosuppressive or cancer treatment are much less affected by COVID-19 than adults. Compared with adults, children recently infected with SARS-CoV-2 have also been found to have higher levels of activated neutrophils, cells that are on the front line in the response to unfamiliar invaders. Neutrophils ingest viral particles before they have a chance to replicate. Children have high levels of signaling proteins such as interferon 1 and interleukin 17 and higher expression of the genes that encoding MDA5, a receptor known to recognize SARS-CoV-2. Epithelial cells that line the insides of the nose could also be coordinating the quick response. After spotting the viral intruder, these cells immediately trigger the production of interferons. In children, these are flush with receptors like MDA5 that can recognize molecules commonly found in pathogens. ACE2 is the main receptor for the entry of SARS-CoV-2 into human cells. The expression of ACE2 in nasal and lung epithelium increases during childhood and further during adulthood. Children have fewer ACE2 receptors with a lower affinity for SARS-CoV-2 and a different distribution across body sites, making the entry of SARS-CoV-2 into cells more difficult. Children with COVID-19 have lower neutralizing antibody levels and less ADE. Pre-existing non-neutralizing antibodies can bind to virions, which can then more easily enter macrophages and granulocytic cells, a mechanism called antibody-dependent enhancement. Such virions covered in antibodies can also more easily replicate, leading to higher viral loads. Children with COVID-19 have lower neutralizing antibody levels, less ADE than adults, and have a less robust T cell responses to the spike protein. Mechanisms by which vitamin D might protect against respiratory viruses include increasing viral killing, reducing synthesis of pro inflammatory cytokines, and protecting the integrity of tight junctions, thereby preventing infiltration of immune cells into lungs. In vitro studies show that calcitriol, the active form of vitamin D, has antiviral activity against SARS CoV 2. Low vitamin D levels lead to higher RAS activity and higher angiotensin II concentration. SARS-CoV-2 can infect endothelial cells and cause vasculitis, activation of coagulation pathways, and formation of microthrombi as a result of endothelial damage, as well as angiogenesis play an important part in the pathogenesis of COVID-19 and can lead to thrombotic complications such as heart attacks and strokes. The endothelium in children is less pre-damaged compared with adults, and the coagulation system also differs, which makes children less prone to abnormal clotting. Though vaccination and masking is the best strategy to stay away from severity of COVID, and nothing could be completely ruled out for this disease, but those extra factors give healthy little or younger ones some extra protection, which is a little piece for the parents of little or younger ones. Thanks for watching. See you next time with another interesting topic. Until then, be happy, be fit, and keep listening to Biotech Treasures.